Topic Notes 4.3 Marine Productivity. So you might want to question, why am I showing a picture of whales? These happen to be humpback whales that I got a chance to see in Juneau, Alaska. And if you look on the picture on the left side, you're going to see sort of a bunch of whale parts sticking out in the water. Most of those are mouths. These whales are feeding. They're feeding on plankton. Of course, we already talked a little bit about what plankton is. Now, the reason why they're actually off the coast of Alaska, basically whales generally migrate into the northern reaches, into the colder waters during the summer. And they're doing that because the plankton is there, because the nutrients are there, and there's more productivity. And that's where they're going to find their food. In the tropics, where they tend to spend the winter, there's really not a whole lot of nutrients and there's not a lot of productivity, so there's not a lot of plankton. So this forms the basis of what we're going to talk about today. So let's get started. So the main idea here is that energy flows and transfers through the marine food web is governed by the laws of thermodynamics and controlled by the productivity of any given system. So we're going to be focusing on sort of the back half of that today in this lecture, that whole productivity component. Here are your learning goals, and again, make sure you're paying attention to them, especially in this longer note set. They're going to help guide you as to what you really need to focus on. All right, let's move on. So we'll start with some basic terms. Biomass and standing crop. So when we're talking about biomass, we're talking about the living mass of organisms. Now, oftentimes when we calculate these things, we tend to look at it as a dry mass because, of course, every living thing, including yourself, you're full of water. Well, in the ocean, it's the same way, in fact, even more so. So we're talking about just the non-water part is the biomass we're really trying to get into. And of course, the standing crop, that would be the total amount of biomass present given a particular volume of water that we're going to be looking at. And that is difficult to calculate to some extent. Like we mentioned, the whole dry mass thing, it's kind of hard to take every herring in the ocean and dry it out and figure out how much it weighs. So we tend to work on estimates. So now some more terms, lots of terms today. Primary productivity. The straight up definition, we're talking about autotrophs that convert sunlight into stored energy. So primary productivity is that process and the measure of how much of that energy we have available. And so we can look at that in a couple of ways. We can look at it as gross and as net primary productivity. So here we go. Gross primary productivity, GPP, is the total gain in energy or biomass per unit area per unit time by autotrophs. Now, why we say biomass is because it's oftentimes very difficult to calculate it in terms of energies, which can be done as like kilocalories per unit, whatever. Biomass is a little bit easier to understand, and it's a little bit easier to compare. And it roughly tells us the same thing. So usually when we talk primary productivity, we tend to talk in terms of biomass. So that's gross. Now, net primary productivity would be that same gain of energy to a certain extent, except we're going to have to deduct from that respiration. Basically, remember, plants are going through photosynthesis and producing these energy molecules, not just for the fun of it. They're doing it because they need food, and they're using that to go through cellular respiration. So they are using some of that in the process. Whatever is left over is your net primary productivity, and that leftover energy or biomass is available to the next trophic level to be able to consume. So we have lots of diagrams, graphs, and numbers today, but it really hopefully will help it all make sense. And we can take primary productivity and break it down into this equation. NPP equals GPP minus R, which is respiration. So that's net primary productivity equals gross primary productivity minus respiration. So if you look at this from a standpoint of an ecosystem and where everything is going, That's what this diagram on this particular slide is showing us. So we'll start with the phytoplankton. Now they're obviously uptaking nutrients from the surrounding water, and they're taking sunlight, and they're producing. They're going through photosynthesis, and they're making biomass and energy molecules. So they have a certain amount of gross primary productivity going on in that green box, that phytoplankton box. Now some of that is going back into autotrophic respiration. You can see that little green arrow looping back within the green box. That's autotrophic respiration. Now from there, you have net primary productivity. Basically, the what's left over after respiration is being sent out of the green box, and it is going into the little brown box called zooplankton and bacteria. So they're grazing, basically. You have your little copepods grazing on all the little phytoplankton everywhere. Now, of course, they're now gaining energy and hopefully biomass too. But notice there's that little red arrow going out of the zooplankton box, that little brown box. That's heterotrophic respiration. They're using some of that energy, and that's coming out of the box. Also understand that zooplankton and most other animals do give out waste products, which tend to be nitrogens in nature, and that's where the nutrient cycling goes back in, because that's going to directly release nutrients back into the water that phytoplankton can then use and consume again. But then you have what's called NEP, or the net ecosystem production. That's basically what's now available to the rest of the various trophic levels beyond zooplankton. That's available to, trophic levels 3, 4, 5, 6, whatever, the whole shebang. Now, most of this net ecosystem production is going to end in some sort of waste product where the animal dies, and whether it's zooplankton, whether it's a fish or a whale carcass or whatever the case may be. And unfortunately, not unfortunately, just the way it works, a lot of that productivity ends up falling into the deep ocean out of circulation in the surface waters. That's one of the kind of key components that we're going to get back to later. But whenever it can be the nutrients after these animals and bodies and such have been kind of decomposed and go through the nitrogen cycle, and the bacteria re-release that as nitrates and phosphates and things that they need, then it then gets back to the phytoplankton and starts the whole cycle over again. We're really honestly looking at the nitrogen cycle here and phosphate cycles and things like that, but we're calling it gross primary productivity and net primary productivity and all that. So we'll get back to that. So let's move on.
So overall, an ecosystem's NPP is the rate at which plants accumulate dry mass, that actual plant material, and of course what is then able to be passed on to other trophic levels. In the ocean, we express this in grams of carbon per square meter per year. And that biomass then that is created is available to that next consumer level. The diagram you're seeing here is showing the entire ocean and all of the net primary productivity basically in grams of carbon per meter square per year. And what I want you to notice is how much of the ocean has very little primary productivity going on. Those are the dark blues, and that spread throughout most of the middle of the ocean basins. That's the majority of the planet. Where you see the reds and the yellows, those are areas where nutrients are available, and we're going to be looking at how that works. But don't let it deceive you. Even those large areas, expanses of low productivity, they still have some productivity. And because those areas are so large, that still plays into the amount of oxygen, for example, that gets diffused back into the atmosphere. So it is still a key component to our global systems.
So here we're going to start some problems. So a little bit of math, but it's really easy. Here's the problem. The net primary productivity off the coast of Peru is 300 grams of carbon per meter square per year. So approximately 120 grams of carbon per meter square per year is lost through respiration. What's the GPP, the gross primary productivity? So you basically just plug in the numbers, do a little algebra, and it should come out to about 420 grams of carbon per meter square per year. Not very hard. Now, similarly, I could ask you for the GPP, which would be slightly easier, or I could ask you for respiration if I gave you MPP and GPP. Kind of all depends. But either way, this is not tough math. You should be able to handle this. Make sure you always keep your units, however, throughout all of when you put your answers in place, because units are really important in science, remember.
The term secondary production is really the same concept as primary production. It simply is dealing with heterotrophs instead of autotrophs. So it's the rate of production of new biomass by consumers using energy gained by producers. Because remember, consumers, secondary level up, they all produce biomass just like autotrophs do. So now let's look at various methods that we can use to measure productivity. One of the ways that we can at least see what's going on in terms of productivity is to measure the amount of oxygen or carbon dioxide concentrations in the water. When photosynthesis is taking place, concentrations of carbon dioxide will decrease, where oxygen concentrations are going to increase because, of course, oxygen is a byproduct of photosynthesis. Because the majority of marine producers are single-celled phytoplankton, there's an easy way to actually mimic this in a closed system and calculate things like MPP, respiration, and GPP. Think of it this way. If a closed bottle is placed in the light, both photosynthesis and respiration will be taking place. If a similar bottle is placed in the dark, there will be no photosynthesis, but respiration will continue. And if you assume that the rate of respiration remains relatively constant, you can compare the readings of both bottles kept in light and the dark to work out the rate of photosynthesis. That's what this diagram is basically showing you, and it's giving you some examples of results that you would have. Using a dissolved oxygen sensor, you would need to take the initial reading before the experiment begins. You'd have to have a reading in the light bottle at the end of the experiment and a reading in the dark bottle at the end of the experiment. In this setup, in this example, MPP ends up being about 10 milligrams of dissolved oxygen. Respiration ends up being about 5 milligrams, and GPP ends up being thus 15 milligrams. Now, another method would be to look at the change in biomass, and we can do this by measuring the rate of accumulation of biomass per unit area. We can do this in several different ways. Generally, it tends to be done through harvesting producers after a set amount of time, drying them out. Remember, we need to get a dry weight that reduces that variation in water content, and then compare those masses. This only measures NPP, or net primary productivity, as the plants will continue to respire throughout the experiment. So it doesn't also account for biomass consumed by herbivores. So you have to consider this as well, especially if you're dealing with field work rather than a laboratory controlled setup. Now, through the advance of technology, satellite imagery has really been able to push the envelope here. We can use satellites to indicate the amount of chlorophyll content in shallow depths of water. That means we can monitor productivity on a relatively large scale globally simply by using satellites. Now, when you're measuring productivity, especially in a vertical water column, you're generally going to have something called the compensation depth, which is the point at which the rate of respiration and photosynthesis is equal. And you can kind of tell that in this diagram. The red line is indicating productivity or production. Respiration is the blue line, and as they cross, you have that compensation depth. Now, if you look on the right side of this graph, you'll notice the available sunlight, and that obviously is going to play into where that compensation depth is. Now, remember back to notes 4.1, energetic submarine ecosystems, we talked about the factors that affect photosynthesis in the ocean, everything from temperature, nutrients, light, and density. So those factors also play into, obviously, productivity, because we're sort of talking about the same thing. And from that point, we can discuss the influences of productivity on the food chain. Obviously, the higher the productivity you have in a particular area, that's going to lead to more abundant populations of consumer or larger food chains, more trophic levels, and more complexity. And when we talk back to those factors that influence photosynthesis and productivity in general, really nutrients and light are the key things that you're going to hone in on.
So now we get to nutrients. All right. So nutrients are in the form of nitrogen, nitrates, nitrites, and ammoniums, and phosphorus, phosphate generally, the phosphate ion. Now, oceans, as we mentioned before, they're kind of like big deserts, and this is a good example of that. This is ocean phosphorus concentrations, and so the violet, purple, pink color basically is like zero phosphorus. So you can see that huge swaths of the ocean don't have it. And if you need that to photosynthesize and go through primary productivity, then that's not going to happen for you. And if you remember the image I showed you towards the beginning of the lecture, where the primary productivities is around the world, you'll notice that they match with this phosphorus graph pretty well. So like I referenced before, most of the nutrients can be found at depth, below actually the pycnocline. Remember when I talked to you about temperature and I said, well, temperature does have something to play with productivity beyond just being temperature. Well, here's where it comes into play. When we talked about salinity, we talked about temperature and density of water, and we talked about the thermocline and eventually the pycnocline. This is a big factor in what's going on here. So surface waters are generally limited unless you mix the layers. But, for example, in the tropics, the temperature difference between the surface waters and the deeper waters is pretty significant, and there's basically a density difference going on there, and so there's not a lot of mixing. So the nutrients that are in the deeper water are trapped, and that's key. If you look at the graph over on the right, you'll notice a few things. The purple line represents the nitrogen concentration. Now notice how it's really low in the surface waters, and then as you start to get towards 100 meters, it starts to get higher, and it continues to get higher gradually as you go deeper. Now cross that with the green line, which is chlorophyll. Now chlorophyll, by measuring the amount of chlorophyll in the water, you kind of have a basic idea of what's going on in terms of productivity, because that's of course where photosynthesis is happening. You notice that the chlorophyll is relatively low in surface waters, rarely at the surface, but right when the nutrients start to come, but there's still a little bit of light, you get a spike in it. So this is showing you this difference between the surface waters and the deeper waters. And if you have mixing, you bring those nutrients up and you have more productivity in the surface waters. If you have a really significant pycnocline, you trap those nutrients deeper in the ocean. Now, in temperate zones, mixing tends to occur in the winter. This is because the temperature obviously decreases in the surface waters, and thus there's not as big of a difference between deep and surface waters, and so you have the opportunity for mixing and nutrients to come up. Now, in polar regions, this mixing can occur more year-round because there really isn't as much of a difference anytime during the year between the surface waters and deep waters. So here's another way to look at it. This is showing phytoplankton and zooplankton in seasonal cycles in the Arctic, temperate, and tropical zones. And you'll notice these, how they chase each other. Phytoplankton, which is the red line, goes up, then followed by zooplankton. Then, of course, once phytoplankton plummets, the zooplankton plummet. There's definitely this cycle that goes on, very much like what we see on terrestrial habitats, just on a slightly different schedule. So where do we get sources of deep water? How do we actually get mixing to occur? Because just because it can mix doesn't mean it's going to without some sort of provocation, with some sort of force intervening. Wind tends to be that force. Wind moving across the surface layers of the ocean will cause surface currents and can cause mixing. There's two basic kinds that we'll talk about for today. One is upwelling. Now this tends to occur along coastlines where you have winds that move away from the shore. And as it does that, it pushes water out. As water moves away from the shore, it creates basically an opportunity for the deep water to come up and replace it. And that creates upwelling. This is where global wind patterns and overall climate do play roles into ocean currents, just like ocean currents and ocean compositions play a role in climate and atmospheric processes as well. There's definitely, it goes both ways. The other possibility is what we call divergent currents. Now this tends to happen around the equator, where you have, due to Coriolis effect and in the way the wind patterns are set up, you have two currents going in separate directions, and that pulling apart again allows an opportunity for upwelling to come from deeper in the ocean. And anytime you have that upwelling, you're basically releasing all those trapped nutrients from the deeper ocean.
Of course, you can have too much of a good thing. Excessive levels of nutrients can lead to rapid increase in algal populations, known as an algal bloom. As algae cells die, bacteria populations increase to decompose all of that biomass. This cycle leads to a hypoxic condition with low oxygen, and that usually is detrimental to a lot of the aquatic life that lives in the area. Some algal species will actually produce toxins as well. This is how red tide events occur. Now, although eutrophication can occur naturally, we are seeing many more instances globally related to human development, and this can be anything from excess nutrients coming from runoff through agricultural fields, urban areas, septic systems, you name it. Here in Florida, we've of course been having a lot of these types of issues related to our Everglades watershed system, in which a lot of the water which used to flow into the Everglades now is sort of stuck in Lake Okeechobee, and the lake has to be managed for depth, and thus a lot of those water releases go both to the east and west coasts of Florida, and they carry with them a lot of nutrients. And that is becoming a big problem, especially here on the east coast in the Indian River Lagoon around Port St. Lucie, and even in the Lake Worth Lagoon occasionally as well. Unfortunately, there are no real simple answers to this circumstance, simply because we have a large population here and a lot of various needs and water demands. So I highly recommend you reading about it and learning about it, because it is affecting your local area. Now we're going to turn ourselves to the efficiency of energy transfers through these systems. So if you think about it globally, only actually a small part of the radiation that comes from the sun actually is fixed by producers. In fact, it's about 0.06%, which, if you were considering efficiency, is pretty low. Photosynthesis itself is also not completely energy efficient. There's a lot of energy that's lost as heat through the process. However, producers need some carbohydrates that they produce, of course, through photosynthesis for their own respiration. So only that NPP, that net primary productivity, is going to move on. When we look at energy transfers beyond the primary consumers, when we go into secondary production, we can use a little bit of an equation to kind of help us grasp what's going on. Now, in this case, C represents the energy consumed. Also, consider this the biomass from the producers that's available. So whatever animal you're talking about will be consuming that energy from the producers. Now, on the other side of the equal sign, we are looking at where all that energy goes. P is the energy available for new biomass production. R is the energy lost through respiration by the consumer. F is basically lost through feces, and U lost through urine. So if you look at it this way, you have to subtract R, F, and U to determine the energy available for new biomass production in that particular secondary trophic level on a particular animal that you might be looking at. So in order to calculate efficiency, we generally express this as a percentage, and it's a really simple equation. You have at the top here the energy transferred to the new trophic level, however much that is, divided by the energy in the previous trophic level. You multiply that by 100, and you get the efficiency in a percentage. So let's give this an actual numeric example. So in this particular example, we're talking about the transfer of energy from phytoplankton to zooplankton. So the phytoplankton captures 153,000 kilojoules per meter per square per year, and that is passed on to the zooplankton, but only 12,856 kilojoules per meter per square per year is passed on to the zooplankton. So the efficiency can be calculated thus. 12,856 divided by 153,000 multiplied by 100 gives you a percent of 8.4%. So that means that the efficiency of transfer is about 8.4%. Now remember, we also talked about the fact that generally these transfers are around 10%, so this works out. So again, as we mentioned, the typical efficiency of these transfers is around 10%. It can be higher, it can be lower, and that's based on what type of conditions you have going on. How much food is eaten, how easy it is for a consumer to digest and assimilate the nutrients, how much energy is used for movement or life functions, how much is lost as waste through metabolic processes. And just remember that this lack of energy in the upper trophic levels is going to mean smaller populations for those larger predatory animals. An example of how this sort of works out, if you think about fish, most fish are ectothermic, which means they tend to mimic the temperature of the surrounding water. This requires less energy to maintain a body temperature because they don't have to. Endothermic fish, like the great white shark, tends to have to maintain that higher temperature. Therefore, ectothermic fish are going to have more energy available to predators than an endothermic fish would.
All right, so now let's start representing some of this stuff visually. We use what we call ecological pyramids, and you probably have seen them before. You might just not have realized it. They're graphical models that basically show these quantitative differences between the various amounts of living material stored at each of the different trophic levels. And they're standing crop. And we can use them in various different ways depending on the data we want to represent. So let's jump into it and see what we got. So we'll start with the pyramid of numbers because it's kind of the easiest to conceptualize. Basically, again, these pyramids end up being a bar graph of some sort, if you will, that shows the various sizes of the various trophic levels. And in this case, we're looking specifically at the number of organisms. So for example, and the one at the top here, the pyramid at the top, there's 11,977 aquatic plants. Somebody actually went out and counted. Maybe this is a lake somewhere, I don't know. There's something like 1,800 something cadfly larvae, 316 bluegill fish, and 13 largemouth bass. Okay, so this is representing basically 4 trophic levels in an aquatic system, and their actual numbers of those organisms. Now, you might notice that this might not go right if you're in certain circumstances in terms of the way the pyramid looks. The pyramid on the bottom is a little bit inverted. Now, this might be a forest where the producers, maybe there's 25 trees. Well, that really isn't a representation very well of what's going on because trees are, well, rather large. They have lots of leaves and biomass. So the number of trees, of course, there's probably more than the number of, whatever might be there, a deer or something wandering around the forest. But that doesn't mean that they don't have a high amount of productivity. This is one of the reasons why pyramids and numbers, they're sort of easy to understand, but they're not particularly accurate. Now, in the ocean, because we're dealing with plankton, for heaven's sakes, can you imagine? Let's count all the phytoplankton in this particular whatever. That's not really possible. It's not really feasible. So you end up having to do some sort of estimation when you do pyramids of numbers, unless you have a real finite ecosystem you're studying. So now let's go to the pyramid of biomass. Now this is a very common representation that we see, and it literally is the mass, usually in grams, of the organisms in a particular level. This is much more accurate and a little bit more straightforward in terms of what the actual data is showing you in terms of trends and whatnot. It does tend to rely on samples that are dry massed, and thus, because you can't necessarily dry mass all of the shrimp in a lake, for example, you have to do a certain amount of estimation to be able to get those numbers, and there's statistics to help you with that. But it definitely is a little bit better. But of course, it does leave open some error as well. Now, the pyramid of energy is pretty much the most accurate you can have because you're actually getting down into the actual energy, the kilocalories per meter square per year of particular habitat. Of course, it is also the most difficult to calculate because how do you actually get that? Well, there's ways to actually measure calories in a particular unit of mass and whatnot, but it is very difficult. But it is extremely accurate to be able to deal with. Now, what we're going to deal with mostly generally is going to be biomass because it's more user-friendly for our purposes in our labs and such.
All right, I know this was a long set of notes, but it's time to turn it back to you. Your in-depth question is explain why energy transfers between trophic levels are not very efficient, around 10%. Make sure to put that in your note summaries and keep thinking.